Eric Davidson. Eric is currently the president of the Woods Hole Research Center. He got a Ph.D. from North Carolina State University in forestry. And he has been a Bay Area resident occasionally. He did a postdoc with Mary Firestone in Berkeley, and also worked in NASA Ames as well. Did some postdoc work there. So Eric today is he's a fellow of AAAS and is currently the president of the Biogeosciences section of AGU, and also the coordinator of the North American Center for the International Nitrogen Initiative, and is a recognized world leader in global nitrogen cycling and ecosystem processes. So today Eric is going to talk to us about the brevity in soil models, and he's going to convince us that this is important, but that also that fine-scale microbial mechanisms are also important. Right.
Okay, I guess that's what I'm supposed to do. Thank you all for inviting me here. It's a pleasure to come back. I always enjoy coming back to the Bay Area. And I'm going to, blame all the blame for trying to encapsulate models into something really short and sweet. I'm going to lay on Mary Firestone, because we got started on that a couple of decades ago in something we called the hole-in-the-pipe model, that we got a lot of traction out of that over the years. I'm not a modeler. I'm not one who develops, certainly not developing code, and not an expert on how one puts together models. But what I want to try to do today is to talk a bit about concepts of how what we need to start with to constructing models to represent very complex processes, such as decomposition of soil organic matter in soils. I'm not sure exactly where all of you come from in terms of your background, and so I'm going to go over some things as the way of background that probably most of you know forwards and backwards, so bear with me a little bit. But I want to make sure I give a little broad context to this before I go more deeply into some of these issues. Let's see if I can figure out. So first of all, we know that there's a heck of a lot of carbon stored in soils throughout the world. The number has been changing a lot over the years. I mean, it's not really changing, but our estimates of the amount have been changing, which shows how poorly constrained our measurements and estimates are. When I first started working in this area, the estimate was that soils contain something like 1,500 petagrams of carbon. To put that in perspective, the atmosphere contains, CO2 in the atmosphere is around 700 to 800, I believe, petagrams of carbon. The carbon in above ground biomass, another very poorly constrained estimate, is probably somewhere around 400 to 500 petagrams. So already what's in the soil clearly is multiples of that. And more recently, those estimates have gone up as there's been more data from sampling deeper in the soils, more data from sampling permafrost and wetlands, and so on. But as we look at the distribution of a global map of what we think, at least some of the estimates that exist of how much carbon is present in soils, this is carbon density in soils, we can obviously come up with some broad generalizations that where plant productivity is low in desert regions, there's very little soil carbon. So probably inputs have something to do with it. And where it's higher in tropical forests, we have higher levels of soil carbon. So inputs have something to do with it. But that's not the whole story, because we have even higher levels of soil carbon in places, in particularly high latitude, obviously the northern hemisphere, because there's more land at high latitude there. And so it's not just productivity, but it must have something to do with climate that affects decomposition. So where it's wet and cool, carbon stocks tend to accumulate. This was quite eloquently described many decades ago by Hans Jenny, looking across the United States, the Midwest of the United States, where you have a nice climate gradient of showing that there is increasing mean annual precipitation coming from the Rockies going eastward towards the Mississippi River. And at the same time, there's a gradient of mean annual temperature decreasing as you go north. And the combination of those two things means that down here in the southwest, you have very low carbon contents in the soils, again, low productivity, high temperatures for perhaps high turnover rates, and higher carbon densities up in the northeast, where you have sufficient precipitation for more productivity, lower temperatures perhaps for lower decomposition rates. So we get an idea that climate is involved in both the production and decomposition of carbon in ecosystems, terrestrial ecosystems. So I just referred to those climate gradients. Of course, there are a lot of other things that affect it, such as carbon dioxide, site fertility, hydrology, the legacy of species composition. And likewise, in the output side, whether we're talking about CO2 or methane, all those climate factors as they're expressed through the soil, such things as fire, we often forget to put things like that into models, and properties of the soils and the actors, the microorganisms that are present there, all affect those outputs. So climate obviously is one of those factors, and the effects of temperature have been expressed in various different ways over the years. The Van't Hoff equation is a fairly simple exponential one from which one can calculate a Q10, the Q10 being defined as the ratio of the process at one temperature and 10 degrees higher, such as this expression of Q10. We'll come back to the Arrhenius function a little bit later. I just wanted to show that there are various ways that temperature functions have been expressed in the past. Now, is this really all that important from a global perspective of whether we should be worrying about the temperature sensitivity of something like decomposition of organic matter in ecosystems? And this is just a little thought experiment that estimates of global respiration are on the order of 100 petagrams per year, maybe 120, another very poorly constrained estimate. But let's say that the heterotrophic component is 50 for just a nice round number. And if the difference between a Q10 of 2 and a Q10 of 5 with a 1 degree increase, in other words, a tenth of a 10-degree change, the difference in respiration, all other things being equal, would be nearly a petagram of carbon. So knowing the temperature sensitivity really does make a difference. A petagram of carbon, right now the anthropogenic emissions are somewhere around 10. So this change would be an important feedback. But of course that assumes that substrate is not limiting, which most likely is not the case. And if substrate is limiting, then really the higher Q10 just simply means you get to that limitation faster. So this alone is not necessarily meaning that understanding this temperature sensitivity is necessarily going to immediately give us an ability to predict what the change in respiration is going to be. But it does show that the magnitudes are big enough that it is worth trying to sort this thing out. Well, as I said, the amount of carbon stored in soils, the estimates have been changing significantly over the years. This is one estimate that came out a few years ago that with now sampling to 3 meters depth in some places across the world, we now have bigger estimates of total soil carbon stocks. Most of it is still in the top meter in some sort of form that cycles rather slowly. But then there's also a great deal in peatlands and permafrost, and more recently another estimate came out for even larger numbers in permafrost, and I think that estimate is we're going to see that going up and down as there's more and more interest going into studying the Arctic and to constraining this estimate. The point I'd like to make here is that if we forget about how big this is and don't include the factors that affect its vulnerability to decomposition with climate change, we're really missing the boat if we're focusing on these other sources instead. Well, the models that have been developed over the years for looking at the dynamics of soil carbon, we have to remember that they were developed primarily for looking at effects of land use change and particularly management in agriculture. And they did a pretty good job of that, such as the Century model and the Rothenstedt-C model. And they conceptually divided soils into the carbon pools into different components that are conceptual components, although sometimes we forget about that and think that they really are real pools that exist out in the real world, and there's some debate about that. But it sort of expresses a continuum, and there's another one out there that is a continuum model as well, that goes from labile substrates to recalcitrant substrates, short residence times to long residence times, maybe some differences in temperature sensitivity among these different pools. But it turns out the problem is that these long residence times can be due to many different factors. It isn't just due to the fact that there's a difference in the plant detrital inputs and that some are more chemically recalcitrant on a molecular scale than are others. That could be one explanation, but it's only one. Probably equally or perhaps more important are physical and chemical protections of the organic matter that renders it inaccessible to enzymatic activity at all temperatures. So the residence time doesn't necessarily work all that well for projecting what the changes are going to be like with temperature changes. I said I'd come back to Arrhenius. He was sort of a postdoc. I don't think they called him that in those days to Van't Hoff, but Van't Hoff was a mentor of his. And basically, it says that temperature sensitivity of a chemical reaction is given by this simple equation in which that exponential term determines for any given temperature the fraction of molecules present with the energies equal to or in excess of the required activation energy. Now, whether this is mechanistic or empirical, I'm not going to argue over that. I thought that at one point, the biologists were applied chemists and physicists, and then I heard a mathematician saying that physicists are really just applied mathematicians. So clearly this has an empirical component to it to be fit. It does have some relation to understanding how reactions work, and so we can think of it in some ways as a mechanistic understanding. One of the things that comes out of that equation is this representation of the gradients of activation energies of molecules. The higher the activation energy, the more energy that's needed for that particular substrate to be worked upon by an enzyme, and temperature. And that this Q10 uses it simply as an index of the effect of a 10-degree change in temperature. Shows that as you get substrates that have higher activation energies for the reaction, you tend to have higher Q10s. Also, you tend to have higher Q10s at lower temperatures than at higher temperatures. So this is something that we can put into equations, and when we can put it into equations, we sometimes get a little carried away in thinking that, well, this is what's most important in models because we can quantify it and we can put it into equations. It probably is important, but I already said those things. Sorry about that. And there is some evidence, in the empirical evidence, that this sort of thing works in some cases, at least. For instance, an example of work of using different substrates added to soils and fitting the Van't Hoff equation and using this B term for the pre-exponential term as an index of availability or degradability of that carbon. The more complex substrates tended to have a higher Q10 than the less complex substrates. Also, long-term incubations of soil in the laboratory. Well, long-term relative to what we normally do in the laboratory. It's not long-term relative to how long carbon stays out there in the real world, and many of the turnover times are in decades or hundreds of years or even millennia. But if you incubate the soil in the laboratory for up to a year at a couple of different temperatures and define these different units, different proportions of the carbon respired, and at the higher temperature, the first little unit comes out much more quickly than at the lower temperature, and so on and so forth, it appears that further on out over that time series, later on in the subsequent additional components of carbon respired tends to have a higher temperature sensitivity than the earlier carbon that is respired, indicating that maybe it's the more labile stuff that has a lower Q10, the more complex stuff has a higher Q10 later in the decomposition process. On the other hand, there's other evidence that point in a different direction from warming experiments, for example. This one at the Harvard Forest showing a change in the carbon release between a heated plot and a disturbance control plot, a plot where they put in the heating cables but didn't turn them on. And at the first few years, there was a rather large increase in CO2, but CO2 emissions declined over time, and eventually there was no change. And this was initially viewed as possibly evidence that maybe the initial substrate was temperature sensitive, and the older substrate that took longer, after that was blown off, so to speak, what was left, that's not temperature sensitive. On the other hand, it just may be that this is substrate limitation, and that the differences that we can see between treatment and control for the decomposition of these things that decompose very slowly, that this isn't a method that we can use to measure that difference. But there have been other explanations as well, such as acclimation of the process. Well, what is acclimation of this process? When you or I move from sea level up to a high altitude before we go climb a mountain, it's a good idea for us to acclimate at that lower oxygen content. That's an individual organism changing its physiology to acclimate. But when we're talking about a process in the soil with a community of organisms, is acclimation really the physiological change of given microorganisms that are already there and remaining, or is it a change in the whole community structure? Or is it a change in the availability of the substrate? So these are additional complicating factors that are difficult for us to figure out how to include those in model presentations. So what do we do? What do we know? First of all, we know that decomposition is an enzymatic process. And another model that we have to work with is the Michaelis-Menten model, which says that the substrate is important under some circumstances, that there is a maximum activity of that enzyme that affects the substrate, but also the affinity of that enzyme, which is represented as the Michaelis-Menten constant, is also important depending on whether the concentration is high or low. And I imagine you've all had that at some point in classes that you took along these lines. And so the characteristics of this is that at high substrate concentration, this Km factor becomes insignificant, and therefore the reaction rate is determined primarily by the Vmax and its kinetics, including its temperature sensitivity. Whereas at low substrate concentrations, the Km does become important, and therefore you need to look at some of the kinetic properties of that as well. There's some evidence that Kms change with temperature. It's not conclusive, but in certain studies it has been shown that that is temperature dependent as well. But in any case, we know that if substrate concentrations are low, that the Arrhenius kinetics of the Vmax, or the temperature sensitivity of the Vmax, may be suppressed. It may not be expressed because of the importance of substrate availability. And in most cases, that's probably what soils are like, because soils are such heterogeneous environments. So, for example, why is it that we would sometimes observe that it seems like the temperature sensitivity declines with drought? So, can we explain that through what I've just presented? Well, as I said, decomposition is mostly an extracellular process. The enzymes either have to diffuse to the substrate, or the substrate has to diffuse to the enzyme or the cell, where the enzyme is. And the diffusivity depends on the thickness of water films. Therefore, if you have a drought, you have thinner water films, lower rates of diffusivity of either enzyme to substrate or substrate to enzyme, and therefore you can have a lower rate of the reaction, which makes it appear that this system is not responding to temperature. So, in this case, because the substrate is important, the Michaelis-Menten kinetics prescribes that there will be lower rates, and we observe an apparent lower temperature sensitivity. So how can we differentiate this and avoid this confusion? One is to first differentiate between what I would call the intrinsic temperature sensitivity, which has to do with the type of molecule, and that which is apparent, which is what we actually are able to observe, given all of the other constraints that occur in the soil. So the intrinsic has to do with the properties of the substrates and the enzymes. The apparent is what we can actually observe when all these other complicating factors are covariating. Ivan Janssens and I, a few years ago, tried to express this in this sort of simple, three-dimensional representation of the apparent temperature sensitivity with this intrinsic sensitivity that I referred to now, recently, about the difference between simple and complex substrates, the difference between high temperatures and low temperatures, and how that affects the temperature sensitivity. But we also showed how there are all these other environmental constraints that can overwhelm this intrinsic temperature sensitivity. Unfortunately, I think the take-home message from our paper tended to focus on this, and that there was a lost message that we intended to emphasize as much or more, that such environmental constraints of the availability of the substrate, whether carbon is sorbed or desorbed, whether it's protected in an aggregate, whether there are aerobic or anaerobic conditions, whether there's water stress, water films affecting diffusion, whether it's frozen or unfrozen. I think that tended to get forgotten, or at least didn't get as much attention as we meant it to get. Some of these, of course, might be step functions rather than a linear function like this, such as going from a frozen to an unfrozen state. So apparent Q10 is what we measure. Is that what we need for projecting changes in the future to, say, changes in climate change? Well, remembering that a simple molecule could be locked inside an aggregate and therefore inaccessible to enzymatic reactions, in that case it would have a Q10 of 1. It wouldn't change if the decomposition wouldn't change if the temperature went up. Even though the intrinsic Q10 of that substrate may be much higher because it's essentially a very simple substrate. On the other hand, the observed apparent Q10, we can't really extrapolate that into the future either, because those conditions, such as in this example, the protection within the aggregate, that too may be changing in response to climate. So, for instance, phase change, permafrost thaw, that makes substrates available. Draining of wetlands, so if hydrology changes as a result of permafrost thaw, for example, that will change the availability of substrates. I already mentioned aggregate formation, destruction, absorption and desorption of mineral surfaces. So if we think of future decomposition and think about what we know we can put into equations from the Arrhenius and Michaelis-Menten equation, this one, the Arrhenius equation here, but we have to remember that it is tempered by the substrate availability, which is really complicated in soils because soils have so many other constraints, such as those that I just presented. Well, that's fine as a conceptual construct, but how do you ever put in these sort of things into a numerical model? And that's what I'd like to present, a way of at least getting a start at that. And I'm amazed to see how a number of groups have taken this and started trying to push it further, which is gratifying. But first of all, we start out with an acknowledgement that the substrate quality does make a difference, at least in terms of Arrhenius kinetics. Whether it's expressed or not isn't always the case, but it's basically there. And anybody who doesn't believe that organic matter, decomposition of organic matter is not sensitive to temperature, try this experiment at home. Go home and unplug your refrigerator. You'll find that in the refrigerator there are a number of substrates that are analogous to what you find in the soil. The milk and the mayonnaise is going to go bad pretty fast, right? But the jar of pickles probably won't, because it's under a very acidic condition that's going to restrict that. The can of beer will probably be fine because it isn't exposed to oxygen until you open it. And then there's my dear mother-in-law's fruit cake, which we tasted after Christmas and put it in the back of the refrigerator and forgot about it, and a year later discovered that the microbes didn't find it any more palatable than we did. That's sort of like humus. So there's a variety of different substrates that respond in different ways to the temperature, and we need to be able to include that. But we need to also separately be able to predict future changes in the substrate concentration. So let's start with the backbone of this Michaelis-Menten equation with two substrates. So I'm, in this case, referring to aerobic processes. But you could change that for an anaerobic process in a different sort of equation that shows oxygen as an inhibitor, a poison to the reaction. And here I'm just representing a substrate, a generic substrate. There could be a family of these equations for different kinds of substrates. Okay? And we know that, as I pointed out, the Vmax is going to be temperature dependent according to an Arrhenius function. And possibly the Kms are also temperature dependent. Maybe according to an Arrhenius function, maybe according to some other function. We don't really know much about that. But we have to consider that the availability of that substrate at the site of the enzyme is really important. So you might, many years ago we had big leaf models when we were first trying to figure out how to represent photosynthesis in global models. We just assumed that there was one big leaf over the globe. Well, this is sort of like a big microsite model, as if there were one big microsite where all of these reactions occurred. But diffusion of that substrate is going to be very important, and availability of that substrate, what of the total carbon in the soil can actually be soluble and diffuse. There are also factors that affect the availability of oxygen, the diffusion of oxygen, such as the porosity and the amount of water that consumes those pore spaces affects the diffusion of oxygen in the soil. So we end up here with basically 7 equations that are linked. And some of them are related to soil properties, such as the bulk density and particle density, the total amount of carbon and how much of that carbon is soluble. Some of it's related to diffusivity processes, physical processes in the soil that are not strictly biological. And then there are these, empirical fittings of the Michaelis-Menten equations that we need to come up with, for which we don't have very good data in many cases in soils. And then there are climate factors. There's temperature. There's soil water content, which of course is affected by precipitation and evapotranspiration. Now, one of the things that falls out of this model mathematically, if we just put in some numbers and get a response surface to it. Whoops, excuse me, clicked the wrong one. Here's respiration. Don't worry about the units. They're relative. Theta is soil moisture and temperature, degrees Kelvin. So as you expect, as temperature goes up, the respiration rate goes up. Right? We put that in the model. It's going to be temperature sensitive. We should expect that. But that's not expressed as much when soil moisture is either so low that diffusion of soluble carbon substrates is limited, or soil moisture is so high that diffusion of oxygen is limited. And so those things are affecting this temperature sensitivity. And it's sort of a Goldilocks, not too high and not too low soil moisture content that gives you the greatest temperature sensitivity. This is not based on any sort of, let me show. This comes back to some, observations, theory, if you want to call it that, going back to the 80s, showing that for activity of aerobic processes in soils, at low water content, you have substrate diffusion. At high water content, you have oxygen diffusion. And at somewhere in between that you have the optimal. In the past, there have been many attempts to fit that sort of a curve empirically using polynomial functions, right? So entirely empirically. The problem is one polynomial function for one soil doesn't work for another soil. This surface is not made by doing polynomial fits, but it is the result of combining the Arrhenius and the Michaelis-Menten kinetics together in those equations. Of course, they still have empirical fits to them, those equations, so we haven't entirely removed ourselves from empiricism, but at least we have a response surface that makes sense in terms of our understanding of Arrhenius and Michaelis-Menten kinetics. And furthermore, if we use Q10 as an index of temperature responsibility, we get what we put in the model. We have the highest temperature sensitivity at lower temperatures, consistent with Arrhenius. But we can have absolutely no temperature sensitivity, Q10 of 1, at very high and very low moisture contents. Well, I just said that. Sorry. Can we test this sort of thing? Well, we can, in the laboratory, of course, we can do enzyme assays. If you do them on soils, you get a good bit of variability from one soil sample to another, so some large error terms. But you can show that we can fit Michaelis-Menten type curves, and we can do this at different temperatures and show that the Vmax is dependent upon temperature. We can compare this to CO2 emissions measured in the field, in this case in a trench plot, so that we could eliminate the complication of root respiration. Although if you do that for many years, you have an additional problem of having changed the substrate inputs. But at least initially, it's a way of looking at, of isolating the responses of heterotrophic respiration. And the gray dots represent measurements from auto chambers that go up and down automatically once every half an hour to measure CO2 efflux from the soil. The black dots are that basically the DAMM model that I just showed you. Obviously, it's not capturing the big spikes, the big ups and downs, which are probably, I would hypothesize, related to release of substrate in wetting and drying. We have changes in soil moisture there affecting diffusion processes, but not necessarily releases of substrate from dead microbial biomass or turnover microbial biomass or desorption, resorption processes. So obviously we're not representing all of the sources of variability on the fine temporal scale. There is some daily diel temperature variability represented. Overall, the seasonal trend is represented. Interestingly, we can get a little bit better fit if we allow one of these terms, which I'll show you again to remind you in the next slide, this alpha term, the pre-exponential term, to vary seasonally. And this is really a way of using a model to create a new hypothesis. So this is that term I was just referring to, that pre-exponential function. If we allow that to vary seasonally, we can do a little bit better. And oh, by the way, the other thing I didn't mention about the model is that it does as well or better as our best empirical fits based on just temperature and moisture content. Okay. So at least we're not doing any worse. And we're probably doing a little bit better. But obviously we aren't capturing all of the sources of variation that we would like to capture. Now, we have the temperature and the moisture content. We can use pure empirical fits with those. But this quasi-mechanistic simple model gets us the same sort of result, a variability relative to temperature and moisture. But what I think we could do for future modules to this, and I'm learning that people are already starting to try to work on this, is that some of these components could be represented by other modules of the model that represent microbial population and enzyme production dynamics. So this pre-exponential factor really could represent, very crudely, changes in microbial biomass, changes in the capacity to produce the enzymes that are involved here. Or properties of those enzymes could be changing in terms of the Km factor, different isoenzymes having different properties. So it too may change in terms of its pre-exponential and activation energies. So that sort of thing could be a completely separate module that fits into this model structure. Likewise, it's silly to think of, very crude, to think of just some proportionality constant of total soil carbon that's soluble. We should be able to do a lot better than that. You could substitute for that something like a century-type model, looking at different fast, slow, passive pools that are turning over and feeding that soluble. Or you could have other types of models, looking at aggregate turnover, sorption and desorption off of mineral surfaces that are controlling the availability of this soluble pool. But that too could be a separate module that fits into this. And also, as I was trying to emphasize before, the importance of permafrost, another, in some areas, the most important thing here might be the rate at which the soil thaws and carbon becomes available. For instance, this recent publication by Jen Hardin and colleagues, trying to improve those estimates, further constrain those estimates of how much carbon is in permafrost. These are units in petagrams, I believe, based on a map of different soil types in the Arctic region, and looking at different horizons and what data sets are available for the amount of carbon and nitrogen in different horizons, and using a couple of climate models and scenarios, these RCPs of high and moderate warming, to try to estimate how much of that total carbon would become available, potentially available due to thaw. And then from that, one has to also consider that some of that carbon may be readily available for decomposition and some may not. But these are the kinds of things that need to fit into that, into the overall model structure, in order to determine the availability of the substrate, to get that into the model as well. So to summarize, by combining these concepts of Arrhenius and Michaelis Menten, we can create a model where we know things are temperature sensitive because it's built into the model. But it's also built into the model in such a way that it isn't always expressed because substrate availability is so important in soils. That may be related to diffusion processes. And so the prescribed temperature sensitivity is fully realized only when substrate availability is high. And apparent temperature sensitivity is reduced when that substrate availability is not high. A modular construct would allow us to make parts of it more complex as we have more understanding. If we can model the dynamics of microbial biomass and enzyme production seasonally or spatially. If we can come up with representations of absorption, desorption, kinetics, and aggregate turnover that can be expressed mathematically as we can currently express Michaelis, Menten, and Arrhenius functions. And this permits both short-term and long-term processes to be used to test the model. I showed you an example of soil respiration, but we could also look at it in terms of longer-term changes in soil carbon stocks. So I think a number of you have probably seen my last slide that I like to show about how difficult it is for us to image what it is that's going on in soils and why it's such a challenge. But I like to show it for those who haven't seen it because it does encapsulate our real challenge of being able to understand these processes. And I thank you for your attention. This one's a bottom-up model, by the way. I'd be happy to entertain any questions. Yeah.
You alluded to the importance of the hydrolytic enzymes in your model, and I'm curious to see how they fit, given that they perhaps don't necessarily have a direct dependence on oxygen.
Oh, well, I mean, you could take the oxygen term out of that. I mean, it's there just to demonstrate that in those cases where it is important for the family, so to speak, of enzymes where it is important, you can put it in, and if it is, you can take it out. And if it's methanogenesis, for example, you'd have to put in an inhibitor effect of oxygen. Yes.
You mentioned, well, you showed two examples which the more recalcitrant organics either more temperature sensitive or less or not at all temperature sensitive relative to more degradable substrates. And I'm just wondering if you have thoughts about what the true situation is likely to be, or is it a really unknown question at this point?
I'm not sure if I understood your question right, but I would say that we know that the enzymatic processes are temperature dependent. Okay, so if we want that, whether that's the most important factor, whether it's so seldomly expressed because other factors are limiting substrate availability, my sense is that that's probably the case, that putting so much emphasis on activation energies is probably not worth our energy. I did make that comparison at the beginning, that if the difference between a Q10 of 2 and 2.5 globally would make a huge difference on the global carbon cycle if substrate availability were not limiting. But I think that's a very big if, and I think to get this right, we have to have the substrate availability issue correct. And we've got to figure out ways to put that into the model. Into models until we do, we can't really answer those questions quantitatively. Mary, uh-oh.
You thought you misrepresented your model. What you just said here was that the critical part of applying carbon is availability of substrate as controlled by diffusion. Yet you've named your model the Dual Arrhenius and Michaelis model. And I asked you, was it simply named that in order to have a DAMM model?
I thought... I thought it was a really damn good name. Well, except that Michaelis Menten kinetics has substrate in two places, the numerator and the denominator. And so if you don't have it in there, you're missing this, you know. The reason that I would defend that as being a damn good idea is that otherwise we're just keep talking about Q10s and temperature responses and activation energies. And this is a way to show how that goes only so far under very limited conditions. And by putting in the substrate, and the best way I could think of putting in the substrate was Michaelis Menten, we change the nature of the dialogue.
So in terms of the drought response, the water control and substrate availability, you might expect to see some sort of a threshold effect, whereas water film thickness decreases, substrate concentration would increase for a period until water film thickness reduces, it impacts diffusion. And in your simulations, there's no evidence of any sort of any thresholds there. Why?
Well, if I remember the physics right, the diffusion is often, or at least in this equation, we use an equation where it's the diffusion is a function of the cube of the water content, whereas concentration, I think, would be a function, just a linear function of the water content, right? So I think it's the diffusion constraint becomes much more severe as you get significant changes in water film thickness. Yes.
So my understanding is that models, we're interested in them because we want to somehow predict the present and the future. And we're able to do that if we can know the present, if we can know what these enzymes, these microbial traits are, what the kinetic parameters are, which we're trying to get our hands around. But we also know that microbes can evolve, and that perhaps some of those parameters have some wiggle room. So I guess my question is, if at one point you were talking about how you could see in a changing environment new enzymes coming into play, enzymes that have perhaps different K and B characteristics. How does evolution perhaps change what we think about as the flexibility of those traits, those enzymatic capacities? If there's a sort of alternative reality that's possible because of rapid microbial evolution, how do we wrap our heads around that?
Okay, well, I think you could use the model to pose that hypothesis. And for instance, in a very crude way, you would say, well, if there's so much redundancy among in the microbial population that this fact, it doesn't, if there's variation, either seasonally or due to whatever time frame that you want to talk about evolution in, and there's so much redundancy that this factor, variation, is not important, then you'd say that if you're trying to use this model to fit your data, you wouldn't need to vary this because it's a constant. You know, there's always somebody out there that can produce that enzyme, and or the enzymes are going to be produced that can always adapt to, or are adapted to, I should say, those environmental conditions at the time. And so the capacity, the enzymatic capacity, isn't an important factor. Okay. Now, we'd made one little attempt to look at variation with, fitting the model in a Bayesian approach. I hope it wasn't Bayesian abuse. And we showed we could get a little bit better fit with varying this seasonally, indicating that maybe in the spring there's more, in a temperate forest, there's more enzyme production, and there's a burst of microbial activity as carbon and nitrogen, other substrates become available. That's a hypothesis. I think there are other tools that could be used to test that more directly. I think testing it with measuring CO2 flux is a pretty crude approach. But you know, the more sophisticated approaches also have ways to go, don't they? to allow them to be used for that. Yeah, go ahead.
Maybe a somewhat theoretical question, but it seems like Michaelis-Menten formulations are really the foundation of your conceptual and numerical modeling. And I'm wondering, just in general, how rigorously that concept has been tested. It's very difficult to test something like that in an extremely complex environment like soil. And we know, studying microbes, pure cultures with single substrates, with the window we've had on this process in the past couple of decades, consistent biology, the genetic regulation is extraordinarily complex. And when you take into account that soils, you have many substrates, you have environmental conditions that are vary. I could go on a long time about the possible factors that could really confound this kind of model. And I mean, how confident are we that this is really the appropriate way, the appropriate foundation for everything built on?
Well, actually, it's interesting. My colleague that did most of the the numerical modeling part of this work sort of got caught up for a long time on the fact that he wasn't satisfied with the Michaelis-Menten model fit. And he started down a road of looking at different formulations that don't strictly follow the classical Michaelis-Menten. And eventually we had to stop that and say, look, we can't answer that question. That it could very well be that there are other kinetics that would fit the data better. We don't know if that means they're more right or wrong. But this is a concept that is, entrenched in our literature and our understanding, and we all learn it in classes that we take as undergraduates. And so my philosophy on this was, let's start with the things that we think we can represent mathematically, knowing that they're not perfect, and they can always be taken out, and you can put in another equation in there if you come up with another one that you think works better. Like another question was asked here, well, what about the case where oxygen isn't important, or what about the case where oxygen is an inhibitor? You can change those. You can make, there may be other factors that are important, that other substrates that are involved, that you can make it more complex. But my philosophy was, let's start with a more simple approach, even if we know in some cases it's, it might be wrong, but it's what we know how to express, and it does a pretty good job. That's just, I guess, the philosophy of starting and not letting the perfect be the enemy of the good. Margaret, you had a question.
Thanks for coming. Just thinking about what we can observe as the apparent Q10. I don't think that, maybe Q10 is kind of a funny variable. If you see an apparent temperature response from an experiment looking seasonally, kind of observation, you think it's always that apparent Q10. Once you've got a micro and a substrate together, you have an environment. It seems like a good set of tools would be to have models where you can learn by trying to fit them to the apparent Q10, to learn about some of the component pieces, and then take those to build a model to look forward.
Well,
I wonder what you think about this, or maybe totally other kinds of models. What do you think about that? How can we, these are observations, what kind of models should we have to go from observations of temperature response back
Well, I think we do that here in terms of, in terms of moisture control. So by putting that in, we test a hypothesis that moisture control, through its effects on diffusions of substrates, changes the temperature response that you measure. And so that's one example where I think that's exactly what we should do. Now, this whole other thing up here, I mean, you could do experiments in which you destroy aggregates and see how that affects your apparent Q10. I would say that you'd probably suddenly observe an apparent increase in temperature sensitivity if you were doing an experiment where you destroyed aggregates and suddenly increased substrate availability. But I think you could construct ways of testing that in a model framework and then making the appropriate measurements in an experimental approach. Yes?
Along those lines, I'd like your example about the refrigerator, but it seems like most of the examples you gave were actually environmental controls. So was the pH of the controls or the lack of oxygen availability?
Except my mother-in-law's fruitcake. That's a substrate control.
So I guess the question that raises for me is how, which along the lines of what Margaret was saying, that there's always an environment and substrate, right? So how do we actually determine these intrinsic defects that are controlled from the substrate?
Oh, that's an interesting question. Well, I mean, you can do that empirically, experimentally as well, under controlled laboratory conditions. And there are some people that think they can do that from first principles of understanding the nature of the molecule. There's also, if you think of, some of these are not just single reactions, but a number of reactions in series. There's been some efforts. I've gone into waded a little bit into another literature looking at calorimetry measurements to see if you can use calorimetry measurements to quantify that, and it's a little hazy to me as to whether that works or not. But it's a good point. It would be useful to have a library that you could look up of the so-called, activation energies of different substrates. Yeah.
Have you considered the priming technique about when you have extra carbon neutral molecules? Does that affect the substrate concentration SX here or change the alpha?
Well, I would say that the way I would hypothesize it is that priming is increasing the microbial biomass and the production of enzymes, and so it would end up affecting this term. But you'd need to have a whole other model to predict it, right?
They consider testing the model in the untrained plot.
Well, in the untrenched plot, the problem there is you've got a whole other, you've got to add a photosynthesis and plant respiration model. Although, I found out somebody is using this to I haven't seen how. It would be interesting to find out. They're using some eddy covariance data and they're incorporating this into some respiration model that they have and comparing it with eddy covariance data from I don't know how they're doing that. I got an email this morning when I was in my hotel, somebody asking me, they'd really like to differentiate between the pre-exponential and post-exponential factors for microbial assimilation of soluble substrates and CO2 production. And they were saying, how do we do that? What's the difference? And I think it was a little circular because they wanted to get at carbon use efficiency. If you knew carbon use efficiency first, then you could partition out the differences between those two. But if you don't know carbon use efficiency, you need to have some sort of other kind of model that's going to tell you what the carbon use efficiency is, so that you would know, it would be in this, this priming type model or microbial dynamics model to know, how much energy is going to enzyme production or producing new biomass as opposed to blowing off extra carbon as CO2.
Just going to go back to the idea of this inherent Vmax idea for substrates. I struggle with this because activation energy is by definition a pathway dependent, not molecule dependent. So if you assign an activation energy to a molecule, you're building in the assumption that of all the possible ways you could ever break down that molecule, they all will fall into a relatively narrow range of activation energies. And just how good of an assumption that is, especially considering we know that certain organisms can outcompete each other on a given substrate, implying that there are really impactful differences in how well organisms can go after a given substrate. So, it's at the assumption of, do we know that all the possible pathways can have the same activation energies?
No, you're probably right, and you throw something else throw something like this out, you find out all the various things, the assumptions that you have to make, and the chinks in the armor. And I'm not trying to say that they don't exist, but that's an interesting question as to whether you. So you have a modeling environment in which you could ask that question: how important? What are the range of different activation energies that you might expect for activity on a given molecule under different conditions? That's what you're saying, right?
Yeah.
So if there are under different conditions, how much would that change the EA? And how important is that relative to some of these other things that are changing in the soil? So at least we could represent it, and we could ask the question, and we could do a thought experiment on it. And then if you're clever enough to figure out how to create a condition where you can actually measure a change, you've actually forced a different activation energy on the decomposition of a particular model, then you could try to create data to compare it to the model output.
Yeah, it's very possible, as you say, that really that's a small factor compared to these other controlling factors that you look at.
There is, for a long time, biologists, not knowing any better, just said as a rule of thumb, biological reactions have a Q10 of 2. And, well, so maybe it's somewhere between 1.5 and 3. Does that really make a difference? I showed that it would make a difference if substrate weren't limiting.
Right.
But if substrate is limiting, maybe it doesn't make that much of a difference.
That's kind of the question I was just imagining when we're trying to get at. Can you think of some practical experiments where you could vary temperature and look at the inherent temperature response and try to see whether or not, just for practical simplification, say the Vmax is attributable to the compound or is it? Seems like it'd be nice to play around and kind of test this. Using a simple model is trying to go back to the inherent temperature response to some of the assumptions.
I hope somebody does it. Yeah.
What's the spatial scale associated with your model? What's the resolution you apply these equations to?
Well, like I said, this is a big microsite model, which is kind of an oxymoron, isn't it? So at the moment, this doesn't really have spatial resolution. And so there are some dirty little secrets here in terms of assumptions of how to get about these diffusivity estimates, and the ones we used in this paper really aren't the right units for diffusivity because we had to make some simplifying assumptions. So that's a whole other area where we need to get some better soil physics in here. And you can ask the question, do we need to, obviously it would be advantageous to represent this in a three-dimensional framework. And then, but on the other hand, you instead, if that's impossible, if that's computationally not possible, you can think of, probability distribution functions of lengths of pathways for diffusion, for substrates to diffuse. So if you can't represent a real three-dimensional soil computationally in your computer, you can simplify it by a diffusion equation where you have to have a distance that your substrate diffuses, but you use a probability distribution function of that distance. So that simplifies it from not needing to be three-dimensional. I don't know. Those are just thoughts.
So this is somehow an upscale form of it's an upscale model, and then you include the smaller scales that you don't capture directly with.
Right. Instead of bottom up, it's sort of top down. We're starting with a backbone of a model, and it can get as much more complex, whether it's different activation energies, whether it's better diffusion models, whether it's better microbial models. We could think of this as like a Christmas tree. You can hang a whole bunch of ornaments on it and feed into it. Okay. Thank you all for all the thought-provoking questions.
So there's a postdoctoral student lunch in room 316, which is directly over here. We've got the chapter very afterwards. And the last thing I'm going to say is before this video goes on YouTube, we'll try and remove references to your mother-in-law.